We are ready to begin the part two of the program. Will everybody please have a seat? So there's something magical about the number three. Religion has its Holy Trinity, literature its Three Musketeers, comedy its Three Stooges, racing its Triple Crown, and the universe its first 3 minutes. And in baseball, it's 3 strikes and you're out. In art, it's the 3-panel triptych, and in science, it's the Nobel Rule of Three. So the original will of Alfred Nobel made no mention of the number of recipients who could share a Nobel Prize. And for the first 50 years of Nobel's, the prize in every category were given to 1 or 2 individuals, with only one exception. And that one exception was the second Nobel Prize in Physics, in which one scientist received the prize, and the other half of the prize was shared by Marie and Pierre Curie. That was the only example of 3 people getting a prize in the first 50 years. The first time a Nobel in medicine was given to 3 people was in 1934 for the discovery of liver therapy for pernicious anemia, and not again until 1945 for the discovery of penicillin. And it was not until 1968 that the Nobel Foundation formally instituted the Rule of Three, decreeing that the maximal number of recipients for any of its prizes would be 3. And following the Nobel footsteps, the Lasker Foundation adopted the Rule of Three in 1997. Science's Rule of Three is reminiscent of art's 3-panel triptych. And the first triptychs go back to the Middle Ages, when Byzantine churches were decorated with biblical altar paintings. Now, the master of the modern triptych is the British artist Francis Bacon, famous for his bold and emotionally charged human images that shock and haunt the viewer, much like the distorted figures of Picasso and Wilhelm de Kooning. From 1944 to 1986, Bacon painted 76 triptychs. His favorite subjects were crucifixions, drinking companions, famous artists, wealthy businessmen, and screaming poets. During the 16th and 17th centuries, poets were celebrated and immortalized in portraits by renowned artists like Raphael, Titian, and Velázquez. And in 1951, Bacon painted 3 papal portraits entitled Pope I, Pope II, and Pope III. But unlike the earlier artists, Bacon depicted his 3 popes not as sanctified figures, but as anti-pope creatures, in which the papal throne becomes the electric chair, the papal attire becomes gaudy dress, and the organ of speech becomes the aperture of a scream. The scream also becomes a double entendre, mimicking the shrieks of viewers who are shocked at what they are seeing. One of Bacon's contemporaries in the London art world was Lucian Freud. And Bacon and Freud had a complicated relationship. They were confidants and rivals at the same time, pushing each other to greater horizons, not unlike scientists who work at the forefront of a new field, as you'll hear about in a few minutes. One of Bacon's most acclaimed triptychs, 3 Studies of Lucian Freud, depicts Freud in a hellish holy trinity, tangled and trapped in a series of cages, sitting like a coiled spring ready to erupt and jump out of the picture frame. In 2014, this triptych was sold at auction at Sotheby's for $142 million, the highest price ever paid for an artwork at auction until last year, when a Picasso painting sold for $165 million. Now, Bacon used the triptych format as a clever way to distill a complex story to its essentials so that it could be painted and told in a bold way. Does the triptych approach for storytelling have any relevance to the rule of three for science. Now, one of the strongest arguments for an upper limit of 3 is that it provides selection committees with strict boundary rules. Selection committees are composed of people with varying degrees of taste, judgment, and decisiveness. And without boundary rules, committees frequently end up including individuals whose contributions are more incremental than original, which dilutes the impact of the risk takers who made the crucial insights. Like a Francis Bacon triptych, great scientific discoveries capture the public's imagination when the discovery can be presented in a defined, engaging way. As you will hear in a moment, this year's Lasker awardees have made discoveries that are original, bold, and adventurous, reminiscent of the triptychs of Francis Bacon. So now to the highlight of our award ceremony, the Lasker Basic Science Award, which will be presented by Michael Brown, who needs no introduction other than to say that he and I have been working together for 43 years, the longest scientific partnership in history.
Thank you, Joe. Let me just say, with regard to Sean's presentation, the only thing better than having that blissful moment of discovery is having somebody to share that blissful moment with. And it's been great to have a scientific partner for 40-some-odd years. So, a false dichotomy plagues American medical schools. The dichotomy states that basic scientists make discoveries and physicians apply discoveries to patients. This year's Lasker Awards expose the fallacy of this dichotomy. The basic award goes to 3 physicians, and the clinical award goes to 3 basic scientists. The physicians discovered a fundamental property of nature, and the basic scientists translated discoveries into a cure for a fatal liver disease. The basic award recognizes 3 physicians who discovered the universal mechanism by which animals respond to a deficiency of oxygen. The 3 doctors are Greg Semenza of Johns Hopkins, Peter Ratcliffe of Oxford, and William Kalin of Harvard. Our cells use oxygen much like a fireplace does. Oxygen combines with carbon to release energy. Cells cannot store oxygen. They require a constant supply. When deprived of oxygen, brain cells and heart cells die within minutes. The result is a stroke or a heart attack. The body needs to sense oxygen deficiency and to correct it. Oxygen is carried by red blood cells. If red blood cells are deficient, oxygen transport fails and cells die. So animals evolved a system to detect oxygen deficiency and to increase red blood cell production. The sensor is located in the kidney. When deprived of oxygen, the kidney secretes a protein called EPO that travels to the bone marrow, where it stimulates production of new red blood cells. EPO was purified in 1977 by the late Eugene Goldwasser. It took him 15 years. With the protein sequence in hand, the gene was cloned in 6 months. The fundamental question was exposed. How do kidney cells activate the APO gene in response to oxygen deficiency? This is not a simple problem. Oxygen is a gas. How do cells measure a gas? And how do they use this information to activate a gene? Mother Nature is a striptease artist. She reveals herself in stages. To expose her beauty, scientists must remove layer after layer. Greg Semenza removed Mother Nature's coat. In the early 90s, he guessed that the EPO gene works like other genes. There must be a protein that activates it. The protein must bind to a piece of DNA near the gene called an enhancer. Semenza identified the enhancer. He used the enhancer as bait to purify the transcription factor, and he named it hypoxia-inducible factor, or HIF. He found that the amount of HIF increases when cells are deprived of oxygen. HIF enters the nucleus where it activates the EPO gene. So Mother Nature's coat was gone. Next came her dress. The riddle was, how does oxygen deprivation increase HIF? This is where our other two winners came in. Working almost in tandem, Ratcliffe and Kalin identified the protein that regulates HIF. The protein has a hybrid German-Swedish name, von Hippel-Lindau. We call it VHL. VHL is a HIF executioner. It binds to HIF and destroys it. When VHL is active, each HIF molecule lives only a few minutes, far too short to activate the EPO gene. Somehow, a deficiency of oxygen must silence VHL so that HIF can survive long enough to enter the nucleus. The discovery of VHL removed Mother Nature's dress. The end was near. But first, Doctors Ratcliffe and Kalin had to figure out how oxygen inactivates VHL. The key is the way in which VHL recognizes HIF. VHL binds to amino acids on HIF that have oxygen atoms attached to them. If these amino acids lack oxygen, VHL can't destroy HIF, and HIF will live to activate EPO. What attaches oxygen to HIF? There must be an enzyme. The race to find the enzyme was on. 3 teams crossed the finish line at once. They were led by Kalin, Ratcliffe, and a late entry into the game, Steve McKnight of UT Southwestern in Dallas. Forgive the plug. The enzymes are called dioxygenases. They latch onto oxygen and transfer it to HIF. Oxygen-coated HIF is recognized by VHL and destroyed. When oxygen levels are low, the dioxygenases have nothing to transfer. HIF lacks oxygen, and it escapes VHL. HIF enters the nucleus and activates the EPO gene. At last, the problem was solved. Mother Nature's beauty was revealed. All great striptease artists save a pleasant surprise for the end, and Mother Nature is no exception. The surprise is a cure for cancer. The search for an oxygen sensor began as a quest for an activator of the EPO gene. Nature's surprise was that HIF activates much more than EPO. It activates 200 other genes. Moreover, HIF is not confined to the kidney. HIF is found in all cells. What other genes does HIF activate? Some of them increase oxygen delivery by stimulating growth of blood vessels. HIF is essential for cancers that need new blood vessels to support growth. HIF also activates genes called cyclins that cause cancer cells to grow. High levels of HIF can be tragic. Rare people inherit mutations in VHL. Without VHL, their cells can't destroy HIF. Unrestrained HIF induces tumors in the eye, in the nervous system, and in the kidney. Even when people don't inherit mutations in VHL, their kidney cells can acquire mutations, and the elevated HIF drives tumor formation. Recently, a biotech company has invented an inhibitor of HIF, and it shows early promise in the treatment of patients with kidney cancer. My striptease analogy is not perfect. Striptease artists remove their own clothes. Mother Nature needs help. Passionate scientists must fumble with the buttons, zippers, and hooks. Doctors Semenza, Ratcliffe, and Kalin did not fumble. Guided by brilliant intuition and powered by delicate technology, they deftly removed each layer. I can only imagine their delight as each discovery moved them closer to the ultimate answer. These 3 doctors exposed the beauty of Mother Nature, and they clearly deserve the Albert and Mary Lasker Basic Science Award.
Well, Mike, thank you so much for revealing those secrets of oxygen sensing. So now, Doctors Kalin, Ratcliffe, and Semenza, if you would please come up here to the podium. The 2016 Albert Lasker Basic Medical Research Award is given to William Kalin, Peter Ratcliffe, and Greg Semenza. Their award is given for the discovery of the pathway by which cells from humans and most animals sense and adapt to changes in oxygen availability, a process essential for survival. Congratulations.
Thank you very much. So I grew up during the space race, and hence I grew up in an era where science and engineering were celebrated and supported, think chemistry sets, microscopes, etc. In high school, I attended an NSF-sponsored summer program for 32 mathematically gifted students that actually changed my life. I was pleasantly surprised to learn that I wasn't the least gifted of the 32 mathematics students, but I had by far the worst grades owing to my abysmal study habits. And so that taught me several things. First of all, school was more fun when I was actually challenged. It's always a good idea to be surrounded by people who are smarter than you are. And I also learned, fortunately, that I too could get good grades if I actually did my homework. As a pre-medical student, I floundered in a research laboratory on an independent study project that was uninteresting, unimportant, and as I would later show, undoable. My mentor rewarded me with a C+ and noted on my college transcript that Mr. Kalin appears to be a bright young man whose future lies outside the laboratory. This convinced me that I was going to be a clinician rather than a scientist, and I approached my clinical training accordingly, and I even served as a chief medical resident at Johns Hopkins, where I honed my knowledge of obscure diseases such as von Hippel-Lindau disease. Years later, fortunately, I discovered, thanks to the outstanding mentorship of David Livingston, that I actually could do science. And my clinical practice taught me that we desperately needed new cancer treatments that would be based on a deeper understanding of cancer pathogenesis. I like math and science and medicine because I like solving puzzles and I like results that are objectively verifiable. Now, as you know, basic science creates or gathers knowledge, and engineering applies that knowledge to useful purposes. For most of my adult life, I've benefited from the wise decision in our country to let the public sector support basic science because the timelines and the deliverables are too unpredictable for investors, and to then let the private sector decide when a line of investigation is mature enough for application and commercialization. And even though I would argue that bargain has served American biomedicine very well, you repeatedly now hear calls to treat science as though it was engineering, tying funding to anticipated outcomes and impact. This is very concerning because you may know that many practices that are useful in managing engineering projects are actually antithetical to good science. And so, for example, building teams and harmonizing goals is very important if you're engaging in a very large and expensive engineering project. But early stage science is often driven by creative individuals who follow their curiosity and have the willingness and ability to go where the science takes them. And I think forcing scientists into teams and also having them answer to predetermined deliverables is actually counterproductive because I think it runs the risk of creating herd mentality and tunnel vision and can stifle the heretical thinking that actually often underlies many transformative discoveries. So JFK knew that it would take approximately a decade to put a man on the moon because at that point this was largely an engineering challenge. It wasn't a scientific challenge. And I think it does scientists, but more importantly, patients and their families a disservice when fundraisers and policymakers oversimplify and overpromise with respect to the great biomedical challenges of our time. I'm very grateful and proud to accept this award on behalf of the many talented young people who've worked in my laboratory over the years, to share it with such esteemed colleagues. And I also wish to dedicate it to my beloved wife, Dr. Carolyn Kalin, who died last year. Thank you very much.
Bill. And now Dr. Ratcliffe.
I am indeed deeply honoured today to receive the Lasker Foundation Award. A moment perhaps to reflect on the twists and turns in life that brought me to this fortunate position. One of the first, rather clear in my mind, dates to the Lancaster Royal Grammar School for Boys, circa 1970. I was a tolerable schoolboy chemist, and following the career of some distant relative, I was keen to study industrial chemistry. One day, the ethereal but formidable headmaster appeared in the chemistry laboratory, summoned me to one side, and said, Ratcliffe, I think you should study medicine, with disarming serenity. I said, yes, sir, with decisively. And without further thought, the university application papers were changed accordingly. To this day, I don't really know whether he felt I would be a good doctor or a bad chemist. But the moment sticks with me as a reminder of the importance of serendipity in a scientific career, at least in mine. I did train in medicine as a kidney specialist and came to this research rather late in my career, fascinated by the extraordinary sensitivity with which the kidneys regulate the hormone erythropoietin to regulate red blood cell production. I felt that the problem was interesting, conceivably tractable, and there was, as you've heard, a new opportunity for study with the cloning of the erythropoietin gene. But some people felt, with the emerging success of recombinant erythropoietin, that understanding how the hormone was regulated was a niche area, unlikely to be of very general importance, and they advised me accordingly to study something else, or this as a sideline if I must. That changed when we tried to use what I thought was a really beautiful method. It doesn't really fit with Mike's striptease analogy. Expression cloning. We want to go straight in, realizing the field's competitive. This means the transfer of the property, oxygen sensing, that's where we wanted to get, from one cell, which made erythropoietin and obviously had it, to another that didn't make erythropoietin and didn't have the oxygen sensing property, or so we thought. And to our astonishment, we found that the recipient cell already had that property. Indeed, every cell that we looked at, to our astonishment, had it. That, of course, destroyed that beautiful expression cloning experiment I so desired, but opened a new horizon, the general importance of this system. So, slightly to my disappointment, we never did, like many in this audience, a beautiful expression cloning experiment. Rather, as Mike outlined, we fumbled our way through the dissecting the various stages with others here and others not here to the oxygen sensor. A beautifully simple system, but every turn was a surprise. The general operation and the mode of operation were entirely unforeseen. Nor did we ever think that the oxygen sensing enzymes would be a classical drug target. So now therapeutic modulation of the oxygen sensing system is a heady and exciting possibility. But it's good to receive this award for basic research, because that's what it is, a piece of animal physiology that, to the best of our knowledge, in its assembled form, evolved about 500 million years ago, a piece of the way we work that was true then, true now, and hopefully will be true in many years to come. That's what's so satisfying. A new medicine is indeed something we'll strive for, respectful of the contributions of not just scientists, but entrepreneurs, business people, so many disciplines that are required to come together to safely develop an effective drug. However, I'm here as a clinician who has enjoyed a career in basic research. A fortunate position, even more so for the valued close colleagues whose names are in the acknowledgements and who contributed so much to the story. Thank you very much.
Thank you, Peter. And now, Dr. Semenza.
Thank you. Many of us who conduct biomedical research do so with what could best be described as a religious fervor. And I think Mary Lasker would approve of that. She once told a reporter, "I am opposed to heart attacks and cancer the way one is opposed to sin." Amen. 7 months ago, Justice Antonin Scalia died. He had a heart attack, which occurs when the flow of blood through one of the coronary arteries is blocked, cutting off the heart muscle supply of oxygen. 8 months ago, David Bowie died of liver cancer. Cancer cells invade surrounding tissue, find their way into blood vessels, and spread throughout the body. What are they searching for? My guess is oxygen. Over the last 25 years, we've tried to understand how a family of proteins, which we called hypoxia-inducible factors, enables cells, tissues, and organ systems to adapt to changes in oxygen availability. As you heard, we started by trying to figure out how the erythropoietin gene is turned on when the body's deprived of oxygen. And now we know of over 2,000 genes that are regulated in a similar manner. Looking forward, I'd like to think that within the next decade, there will be drugs which stimulate the activity of hypoxia-inducible factors that will be used to treat anemia and cardiovascular disease, and drugs that inhibit these factors will be used to prolong the lives of cancer patients. So if you will, this is my religious fervor. I'm filled with wonder at the outcome of 4 billion years of evolution here on our speck in the universe, and hope regarding our opportunity to improve the lives of those around us through basic science discoveries and their translation to clinical practice. Well, I began with Mary Lasker, so I'll end with Albert. As you know, Mr. Lasker was a very successful advertising executive. He was in charge of accounts for products like Sunkissed oranges and Pepsodent toothpaste before he and Mary began their campaign to promote biomedical research. Coincidentally, my grandfather also worked for an advertising agency here in Manhattan. His account was for a product called Wonder Bread. Thank you very much.
Congratulations, Drs. Kalin, Ratcliffe, and Semenza. And now I would like to ask Dr. Harold Varmus to come to the podium.
Wait till it's over, guys. See if you like it. So most of us don't spend too much time thinking about our livers. Unlike our pumping hearts and our reasoning brains, the liver is normally a silent, though essential, partner in our lives. Right now, it is quietly working on our lunch, keeping us in metabolic balance. But when damaged by alcohol, infiltrated by metastatic cancer, or felled by a viral infection, it can no longer be ignored. Hepatitis C virus, or HCV, is one of the things that can bring the liver to our attention. The virus is now growing persistently in the livers of an estimated 170 million people around the world and over 3 million in the US. With time, HCV can destroy the liver's essential functions, since an infection is often a prelude to cirrhosis or to cancer. Today, we celebrate 3 people whose work has enabled medicine to eliminate infection with HCV. To eliminate, not just control, a chronic viral infection is unprecedented. And the story is unusual in other ways as well. First, why that C? In medical school, I learned about hepatitis A, a self-limited viral infection that was usually transmitted by food, and about hepatitis B, a chronic infection usually contracted at birth or by transfusion or intravenous drug use, often causing cirrhosis or cancer. Over the next 20 years, vaccines were developed to prevent these infections, although there are still many millions of people around the world chronically infected with hepatitis B virus. We developed tests for hepatitis B virus that makes transfusion safer. But a big part of the hepatitis story was clearly still missing. Harvey Alter at the NIH Clinical Center showed that another prevalent hepatitis virus, neither A nor B, must be in our blood supply. For example, he could transmit it to non-human primates and cause disease. But the usual methods did not help him to identify the so-called non-A, non-B hepatitis virus. Where traditional virology failed, molecular biology rescued us. In 1989, Michael Houghton and his colleagues at Chiron Corporation isolated a piece of nucleic acid from the blood of an infected patient, a patient infected with non-A, non-B virus. They found that it was part of the genome of a novel virus now called HCV that belongs to a known class of human pathogens. The virus was enough to design a diagnostic test for HCV, protecting the millions who receive transfusions each year. And in 2000, Alter and Houghton received a Lasker Clinical Prize for their work. That fragment of the hepatitis C virus genome could have been the toehold that led quickly to the production of abundant virus and to the design of a vaccine and antiviral drugs. But that didn't happen. It didn't happen because HCV is an odd and perplexing virus. And here is where today's heroes enter the story. Charles Rice had been trained at Caltech, working on the class of viruses, the flaviviruses, to which HCV belongs. So when that HCV fragment had been announced, he saw its potential. By then, he had joined the faculty at Washington University in St. Louis, and his laboratory aimed to do what could be considered the obvious thing, to extend that fragment into a complete viral genome, a starting point for making infectious hepatitis C virus in cultured cells. One could argue it should have worked easily, but it didn't. When he and some others finally found the missing pieces, he learned that variations in HCV genomes impeded the production of virus in cultured liver cells. Finally, he was able to puzzle together a genome that did make virus, and the virus could infect a chimpanzee and cause hepatitis, and this was a major achievement. But that virus still didn't multiply in cultured cells, so essential studies were stymied. Ralph Breitenschlager was working in a parallel universe, trained in Heidelberg, working on the hepatitis B virus. After joining a pharmaceutical company in Germany, Ralph also saw the opportunities that were inherent in Chiron's piece of the HCV genome. And then he experienced many of the same frustrations that Charlie Rice had experienced. And he and Charlie commiserated about these at international meetings for nearly a decade. After returning to the university in Heidelberg, Ralph and one of his trainees, Volker Lohmann, conceived an interesting plan. They would try to multiply only a part of the viral genome in liver cells. Now, the strategy precluded the production of complete infectious virus, but it allowed them to select partial genomes that could multiply efficiently. In turn, those efficient partial genomes allowed drug companies to seek compounds that inhibit the replication of the partial genomes and other viral functions. Now, let's remember that looking for antiviral drugs was not, and still is not, a trivial exercise. Before the development of drugs that inhibit HIV, the human immunodeficiency virus, few antiviral drugs existed. Treatment of viral infections, like HCV infections, if possible at all, usually depended on toxic drugs, like interferons, that bolster host defenses. Work with HIV taught us how to interfere specifically with viral enzymes essential for viral growth. So ambitious chemists got to work on the partial genomes of HCV, and Michael Sophia was among those. In 2005, Mike had just left a large pharmaceutical firm to work at Pharmacet, a much smaller company which sought drugs against a number of viruses, including HCV. One type of compound looked especially promising. It inhibited the viral enzyme that copied the HCV RNA genome, but it didn't behave well in animals, rapidly decaying or failing to get into cells. With a series of chemical tricks, Mike and his team turned it into a drug that was concentrated and then activated in the liver. That drug, now called sofosbuvir, turns out to be highly effective against many different strains of hepatitis C virus, is impervious to drug resistance, and has become the common component of the several drug combinations approved by the FDA over the past 3 years for treatment of HCV infection. These combinations can eliminate HCV from as many as 99% of infected patients over several weeks of treatment with minimal toxicity. Some benefits to patients have been immediate: improved liver function and loss of infectiousness. Others are very likely: the sharply reduced risks of ultimately experiencing cirrhosis or liver cancer. Now, everyone here will recognize that these results are extraordinary, but thus far the number of HCV-infected people has declined only slightly in this country or worldwide, because most have not yet received the drug. One reason, among several, for this is their cost. The public outcry about the cost has not escaped the notice of those of us who sit on the Lasker jury. And we have also noted that routes to wider access are being explored and sometimes used. But our job today is to celebrate those who walked a winding scientific trail that led to the miraculous drugs and to the benefits of using those drugs. At the same time, let us resolve to find the means to provide them to all who can benefit. And let us hope that by making these awards today, we have explained why it is so important to do so. Thank you.
Thank you, Harold. And to further emphasize the power of those comments, I'd like to tell you a story. It's a story about Jim Wilson. Jim is alive today because of the life-saving advances which have been recognized by 2 Lasker Awards. Jim's liver failed from hepatitis C infection. He received a life-saving liver transplant, a procedure honored by the 2012 Lasker. But as happens, the virus came back in his liver and almost killed him. Jim was saved a second time by the drugs developed thanks to today's new laureates. And now he gets to spend time with his grandkids and teach them to surf. And we are delighted that Jim is here celebrating with us today. Jim, will you please stand and be acknowledged by the crowd? and his wife Francine, who's with him. Thank you so much. And now that we've been reminded of the power of the work of today's winners, Drs. Bartenschlager, Rice, and Sophia, will you please come forward? The 2016 Lasker DeBakey Clinical Medical Research Award is given to Ralph Bartenschlager, Charlie Rice, and Michael Sophia for the development of a system to study the replication of the virus that causes hepatitis C, and for use of this system to revolutionize the treatment of this chronic, often lethal disease. Congratulations.
Passion, which most often requires an adequate trigger, supporting people, and luck, to my opinion, are the most important ingredients for a scientific career. And I must say I had the fortune to have all three of them. So when I grew up with my grandparents in a suburban industrial area of Mannheim in Germany, my main interest was playing soccer, and I had no clue what my future profession would be. So this changed at high school, where I met an excellent teacher who introduced me into the topic of biology. And obviously, recognizing my new inspiration, my grandparents bought me a small microscope as a Christmas present, which I used in my spare time to microscope and analyze pretty much everything I could cut and put under this microscope, although most often I didn't see anything because, as I learned later on, the numeric aperture was so lousy. Nevertheless, during those days, I decided to study biology, even though people were telling me during that time, which was in the late 1970s, that the demand for biologists in Germany is pretty low. In fact, one biologist per year. So they asked me, why should I be this single biologist? So nevertheless, my passion for this topic was strong enough to not care about my professional future. And after a 4 years interim period where I was trained and practiced also as a police officer, I started to study biology at Heidelberg University. Now, during my time as a student, I developed a growing interest in molecular biology, which in Germany in those years was at its infancy. However, there were very motivated professors in Heidelberg who had done a postdoc period time here in the US, and they returned back to Germany to build up their laboratories using most modern molecular biology methodologies. One of these labs was a laboratory that was headed by Heinz Schaller, a pioneer in molecular biology and molecular virology. So Heinz worked on hepatitis B virus, which was a topic I was very much fascinated about, a really small and very clever virus. And it was not an easy task to enter the laboratory of Heinz Schaller. It was a multi-step process, but finally I ended up because he accepted me as a lab practical student, and my first job was to develop a method for cloning a series of duck hepatitis B virus mutants. I assume I did my job right because Heinz accepted me as a PhD student, and he took me really under his wings to tell me the secrets of how to work scientifically, to work quantitatively, and do the things right. So I left his lab in 1991, and in 2002 I got the opportunity to return back to Heidelberg thanks to a foundation that Heinz Schaller and his wife Chica Schaller had established, which donated money to implement a professorship for molecular virology. And after an international call, I had the opportunity to get this position, for which, of course, I was and still am very thankful. Now, success in science does not only require passion and supporting people, it also requires luck. And I would say my biggest luck was the privilege to host numerous very talented students and postdocs in my lab. And they all have contributed quite a lot to the success of our work, but there was one person who influenced my scientific career most, who was already named. It was Volker Lohmann, who was my very first PhD student and turned into a long-term scientific collaborator, a good colleague and friend. Now, I would say that recruiting Volker was probably the biggest stroke of luck in my scientific career, not only because he was the key person to set up this HCV replication system in the laboratory, but at the same time, he was also an exceptional mind and a great team player, creating a lot of team spirit within the lab, which was a crucial element for the success of our work. Now, being a scientist, as you may probably imagine, is not just a job, it's a vocation. And the pursuit of knowledge and the elation of discoveries are the twin triggers of passion in science. But this vocation also comes along with some aspects like long working hours, being often away, and hectic days, which are aspects that require a highly sympathetic family. Now, without the continuous encouragement of my wife, Judith, who is also here in the audience and whom I would like to congratulate to her birthday today, and without the enrichment of my children, Marie, Nora, and Lawrence gave to me, and that they have provided to my life, I think for sure I would not be here. Thank you.
Thank you, Rolf. And Dr. Rice.
As I look back over nearly 40 years of studying virus, I certainly never imagined that I would be standing here today. Viruses are incredible teachers, and unraveling their secrets continues to reveal new wonders about biology and evolution. As you've heard from Harold, my interest in flaviviruses was kindled as a postdoc and continued when I began my laboratory at Washington University. We studied the prototype family member, yellow fever virus, a nasty mosquito-borne virus made famous by Walter Reed. It became the poster child for human vaccination based on a live attenuated variant derived in 1937 by Nobel Prize recipient Max Tyler. So I was in the midst of exploring the details of yellow fever replication. Hepatitis C appeared on the scene in 1989 as the major cause, you've heard, of non-A, non-B hepatitis. Given its similarity to yellow fever and other flaviviruses, we began a small effort on this new human virus that was associated with progressive liver disease and cancer. And we really weren't specifically searching for a cure. We were just trying to better understand the virus. Together with Ralph and others in the field, we made progress but struggled, as you've heard, to get this virus to grow in the lab. And with each of these apparent sort of breakthroughs, we were met with new roadblocks to overcome. So really, in both patients and in the laboratory, hepatitis C was really this sort of persistent troublemaker. I have to admit that many of us in the field became increasingly frustrated by the difficulty in working with this virus and our slow progress at a time when new therapies were sorely needed for patients. But when the breakthroughs finally came, the tools in hand to validate potential antivirals and to screen inhibitory compounds, a flood of new drugs rapidly ensued, and you'll hear more about that from Mike's comments. And we now, as you've heard, have regimens with amazing efficacy, very few side effects are now approved in the clinic, although we still face problems in really sort of implementing this therapy on a global scale, which we would all like to see. So it's wonderful to have played a small role in this biomedical success story, and I encourage you to sort of look at the acknowledgments to find out sort of all the wonderful people that I've had the honor to work with over the many years, including Ralph and his lab. But just sort of an anecdote, about 20 years ago I was contacted by a family whose little girl had hepatitis C. And we exchanged emails over the years, and for years they agonized over whether or not to treat the little girl or not. And they decided to wait for these new drugs. And Just last week, this message arrived in my email box, and I'll just read it. It says, "Hi, Charlie. I just read that you will be receiving a Lasker Award for your work in helping cure Hep C. Congratulations. We are so excited for you and remain grateful that our daughter is cured and leading a happy, normal life. In fact, happily married with an 18-month-old, our daughter is now expecting her second child. And I think for basic scientists, there's really no reward that is more unexpected than this or better than this. So in closing, I just want to sort of tell you another anecdote. I remember my mom asking me sort of, why do you work on yellow fever? We have a vaccine for that, don't we? She was right, of course, but we were curious about that virus and just sort of forged ahead anyway. And this experience with yellow fever paved the way for our work on hepatitis C, and to me reinforces the importance of keeping a diverse research portfolio. You really never know what's going to come next. It also underscores the importance of supporting and promoting fundamental curiosity-driven research as the driver, I think, of great medical advances. So I want to thank you all for sharing in this, and I really am sort of honored by this award for me and the field and my colleagues. Thank you.
Thank you, Charlie. And now, Dr. Sophia.
So I came to science pretty early in my life. As a kid growing up in a row home in Northeast Baltimore, I can remember borrowing books from the local Baltimore City Public Library, reading about great scientists, and fascinating about making those next great discoveries myself. We didn't have much. So at Christmas time, my brother, my sister, and I were only allotted a $10 budget to buy gifts. Yes, I was the one who asked for a microscope one year, a telescope the next, and a chemistry set the following year. Mom and Dad were really not highly educated individuals. Dad was an immigrant with an 8th grade education. Mom was a payroll clerk. But even with their modest backgrounds, my parents instilled in us the real value of an education, that immigrant work ethic, and the desire to do something good in life. I was lucky enough to attend the Baltimore Polytechnic Institute Magnet Public High School with some of the brightest students in the city. Case in point, Lasker jury member Jeremy Nathans was my classmate. Many of them smarter than I, but I know I had to work harder than them. As my wife can attest, that work ethic my parents held true and hold true today for me. I guess I feel medical breakthroughs don't just happen. My first experience in a chemistry lab actually began as an undergraduate research assistant in the laboratory of Jerry Meinwald at Cornell. Before starting my first experiment, a senior graduate student pulled me aside and said, "You must understand that in research, only 20% of what you do will ever work, and that you will fail 80% of the time. So you must be satisfied with that 20% success rate. Otherwise, research is not for you." Today, I still remember that moment and think to myself, was he ever the optimist? The development of sofosbuvir can be called a classic in the annals of drug discovery, but it also epitomizes the struggle in the field. Today, no one talks about the failures, the disappointments, the doubters along the way, but there were plenty. No one believed we could develop a potent nucleotide drug that could enter the body and target the liver. Few believed anyone could achieve an interferon-free cure as quickly as we did. But our small group of scientists at this little biotech company made it happen and transformed HCV therapy forever. For someone who craves the idea of building molecules to change the course of human disease and working at the interface of many scientific disciplines, being a drug hunter cannot be more exciting and more noble a pursuit. Drug discovery broadens your horizons, challenges your thinking, and if you're persistent enough, occasionally rewards you with the most gratifying real-world medical breakthroughs. Today, I am frequently asked, what are you going to do now that you have discovered a cure for hepatitis C and been part of a successful biotech? That's usually followed by the suggestion, why don't you retire and sit on a beach somewhere? My reply is that there's still too much more that needs to be done. There are 350 million individuals infected with hepatitis B virus who are begging for a cure and need us to keep trying. What motivates me to keep going? Just listen to the stories of the hundreds of thousands of HCV sufferers like Jim Wilson. Their experience of despair and their ultimate triumph are all the motivation one needs to persevere and continue to do something good in life. I'd like to thank the Lasker Committee for this incredible honor, and certainly my colleagues at Pharmasset who've contributed immensely to this success. Thank you.
Congratulations to the winners of the Lasker DeBakey Clinical Research Award. And now, to describe our final award recipient, Dr. Joe Goldstein.
So our Special Achievement Awardee, Bruce Alberts, is a rare bird. But what type of rare bird? He is clearly not a bald eagle or a crackling goose, but he may be a hybrid of a Pacific loon and a wild turkey. So speaking of turkeys, I think the best way to describe Bruce is to invoke a culinary comparison. Bruce Alberts is the Julia Child of science. Let me explain. Julia Child was a master chef, a best-selling author, skillful teacher, and a passionate educator who touched the lives of millions by helping them discover that cooking can be exciting and fun. A ditto for Bruce Alberts, master biochemist, best-selling author, skillful teacher, and a passionate educator who has touched the lives of millions by helping them discover that science can be exciting and fun. The careers of Julia and Bruce began in much the same way. Both experienced a sudden awakening that changed their lives. Julia fell in love with French food and Bruce with science. Bruce also later fell in love with food, I think. Julia's Epicurean epiphany came at age 35 at a restaurant in Rouen during her first trip to France in 1948. Her first French meal was simple: oysters on the half shell, sole meunière, a green salad, and a chocolate souffle. Until that moment, Julia knew nothing about French cuisine and didn't know the difference between cream of wheat and creme brulee. Her idea of a good meal was a shrimp cocktail, meat and potatoes, and Jell-O for dessert. Now, in a miraculous transformation over the next 12 years, Julia became an authority on French cuisine. In 1961, she published her first book, Mastering the Art of French Cooking, which remains in print to this day in its 18th edition. This book is a classic because it demystified French cooking. French baked beans sounded less mysterious to Americans than cassoulet. In 1962, Julia was invited to appear on Boston Public Television to discuss her new book. She arrived at the TV studio with a dozen eggs, a whisk, and a hot plate, and during the interview, she whipped up an on-air omelet. The audience went wild, and the rest is history. And now to Bruce Alberts, our Julia Child of science. Bruce's true calling, like Julia's, came in a flash while standing in front of a classroom in Glencoe, Illinois. When Bruce was 12, his 7th grade science teacher asked him to explain how a television set works. That was back in 1950 when television was new, and Bruce had no idea how it worked. His scientific epiphany came when he realized that in order to teach how something works, you first had to understand it. Bruce's fascination with science led him to Harvard, where he did graduate work in Paul Doty's laboratory on the physical chemistry of nucleic acids. In 1960, he embarked on a series of ambitious experiments to figure out how a cell replicates its DNA when it divides into 2 daughter cells. The results were equivocal, and to Bruce's consternation, a group of distinguished Harvard professors at the very last moment did not approve his degree. Julia Child suffered a similar fate at the Cordon Bleu cooking school in Paris. In her final exam, one instructor failed her because she didn't know that an œuf mollet was a soft-boiled egg. Undaunted, Julia spent the next several months perfecting her French, retook the exam, and got her diploma. Like Julia, Bruce persevered, did 6 more months of experiments, and got his degree. The traumatic wake-up call on a crimson carpet propelled Bruce to become a great biochemist. In the 1960s, DNA was thought to be replicated by a single enzyme, DNA polymerase. No one guessed that the enzyme would be only one component of an elaborate machine formed from multiple interacting proteins, each with a different function. Bruce developed new affinity chromatography methods for DNA and protein that allowed him to identify and purify the multi-protein machine that governs DNA replication. This work became the prototype for other protein machines in the cell, such as those that splice RNA and secrete hormones. In the 12 years after his Harvard wake-up, Bruce underwent a miraculous transformation. He had now become one of the leading biochemists in the world, and in 1976, he took over the chair of biochemistry at the University of California in San Francisco. Here, he perfected his skills as a teacher, mentored young faculty members, and began to dream up innovative ways to connect working scientists with public school teachers. But probably most important of all, Bruce teamed up with 5 other scientists to write the most influential textbook of its kind, Molecular Biology of the Cell. The first edition was published 32 years ago in 1984 when Bruce was 47 years old, the exact same age as Julia Child when her first book appeared. Like mastering the art of French cooking, Molecular Biology of the Cell has been a dazzling success. It is now in its 6th edition, has been translated into 11 languages, and has been devoured by tens of millions of students, as well as established researchers, all of whom praise it for its clarity, logic of its explanations, and splendid illustrations. Even though the current 6th edition has 7 authors, the material is integrated in such a way that it reads like the work of a single hand, the deft hand of the master chef, Bruce Albert. From 1993 to 2005, Bruce served as president of the U.S. National Academy of Sciences. And like Julia, who used public television as a bully pulpit to raise the food consciousness of the American public and enhance sophistication of their appetites, Bruce used the presidency of the NAS to inform the nation of the importance of science education and to champion the role of scientists in society. Under his 12-year leadership, the NAS and its 2 sister organizations published an unprecedented 200 reports on all aspects of education from kindergarten to graduate school. In his crusade for implementing national standards for education, Bruce urged that schools quit teaching dry and deadening facts that students memorize just to pass tests. According to the gospel of Bruce, students should be taught how to think critically, solve problems rationally, and acquire the good taste needed for a successful career in science. Recall that the Albert's philosophy actually began 66 years ago when a classroom of 12-year-olds in Glencoe, Illinois, were first exposed to the anatomical workings of a television set. Speaking of television, Julia Child literally made public television. She was the first public TV star to become an internationally known figure. Her phenomenal success was the result of 4 things: missionary spirit, boundless curiosity, endearing klutziness, and a deep desire to teach and to teach well. This sounds exactly like the perfect description of this year's Special Achievement Awardee, Bruce Albert. Bruce, congratulations.
Thank you, Joe. I'm sure Julia Child would have been really proud to hear herself compared to the world's master science educator, Dr. Bruce Alberts. Come on up. The 2016 Lasker Koshland Special Achievement Award is given to Bruce Alberts for fundamental discoveries in DNA replication, for visionary leadership in directing scientific organizations, and for passionate dedication to improving education in science and mathematics. Congratulations, Bruce.
Well, thank you, Joe. I especially like the klutzy part. It's a real honor to receive this award, of course, but especially because it's named after Dan Koshland, who was a close friend and an inspiring example of how to be a great scientist and a great scientific leader. Like almost everybody else who's a scientist working on cells, I, of course, read about, taught, wrote about his wonderful scientific breakthroughs. But I had a special opportunity to work with Dan because when his wife, Bunny, Marion Koshland, died, he decided to endow a new science museum at the National Academy of Sciences. And of course, Dan insisted on doing as much of everything himself. So it was Dan and I who were left as a scientist to design the first major exhibit, which was on DNA. And if I go back in the Academy records, there must be over 1,000 emails back and forth because, you know, Dan was a stubborn guy. But in the end, it turned out really well. And Dan had a wonderful sense of humor, which made it all such a wonderful experience for me to work with him on that. So I want to focus in a very brief time I have on my passion today, which is greatly improving, changing the way we teach science to children age 5 through college. And my mission would be to help recruit many more scientists to this effort because it's not going to happen without us. My science education, I really don't mean what happens in most schools today. Memorize all the facts that you can about what scientists have discovered about the world in biology, whatever, chemistry, whatever. That I don't think is important. What I think is important is teaching science as the practice of science. Kids have to experience trying to solve age-appropriate problems in school. And in that way, everybody would hopefully gain critical thinking skills that the world needs, that everybody needs. So basically, I would like to advertise the wonderful 3-minute video that Flora Lichtman made. I talked to her for an hour talking about garbage, and she took that and made 3 minutes out of it. And it actually makes the point much better than I can make today. So I like to emphasize something I discovered when I first came to the Academy and made my first trip to India. Prime Minister Nehru, the first Prime Minister of India, coined the term scientific temper. And he saw very clearly that his nation, as a new democracy with 50 different languages and all kinds of religions and everything else, could not really be a success without science. I just want to quote from what he wrote in 1948. This is sort of his definition of scientific temper. He said, "The adventurous and yet critical temper of science, the search for truth and new knowledge, the refusal to accept anything without testing and trial, the capacity to change previous conclusions in the face of new evidence, the reliance on observed fact and not on preconceived theory. All this is necessary, not merely for the application of science, but for life itself and the solution of its many problems." So he saw a scientific temper as something that had to be imparted to all citizens of his new country. Now, our world since then has become ever more crowded and contentious. More and more powerful weapons, dangerous weapons have been developed. And really, are we going to be able to survive as a society? I seriously worry about it, unless we work really hard to change the way that people think about their fellow human beings. It becomes ever more urgent. So how are we going to do this? So I'm going to tell you my hypothesis. Scientists have hypotheses. We always have a solution for every problem. That's why we're so optimistic. My bottom line is that if we could introduce high-quality, inquiry-based science education at all levels, starting with 5-year-olds through college, and we actually made a science out of this education and see what happens as we do different things. This is a great opportunity for developing a real scientific temper, the kind of scientific temper that's critical for every nation. The good news is all parents should want this for their children because it's exactly the same kind of problem-solving skills, insistence on evidence and logic that is important for anybody to have a good job these days in the modern world of work. So we're not selling scientific temper. We're producing capable human beings who could actually survive in our society and not be fooled and make our world a safer place. So the important thing from a lesson I've learned from all 30 years now I've been working on this, I don't think this will ever happen without the deep, strong, and continuous involvement of the scientific community in every nation. There's actually an international movement of world's academies to work on this, but we're we're maybe 1% of the way there. So I very much welcome this award, I take it both as a statement that this issue is a critical one, this kind of new science education to create a scientific temper, and very importantly, as an acknowledgement of the reality that this mission will never be accomplished without a major mobilization of scientists. Thank you.
Thank you, Bruce. Thank you to all of our winners, and thank you to each of you for joining us here today and for working with us to advance the mission of increasing support for medical research. Have a great day.